Scientists ran a cloning experiment for 20 years straight. They cloned the same mouse over and over again, 58 times. And what they found changes everything we thought we knew about cloning, and maybe even about why sex exists at all. Okay, so let's get into this because it is a wild story. For 2 whole decades, a team of scientists embarked on this truly ambitious quest. They wanted to see if they could clone a mammal, a mouse in this case, forever. This was not just some cloning experiment. It was about pushing the absolute limits of biology itself. I mean, it is a huge question, right? Can you make a perfect copy of a copy of a copy and just keep going without the quality ever degrading? Now, for some plants and simpler animals, sure, that is totally normal. But for mammals, that has always been one of biology's great mysteries. And this experiment was designed to finally get an answer. So, the method they used is called serial recloning. And on paper, it sounds pretty straightforward. You take a regular cell from a cloned mouse, you use its DNA to create a brand new clone, and then you just do it all over again with the new clone. Think of it like a biological photocopier, where each new copy is made from the previous one. Now here's where it gets really interesting. For the first leg of this marathon experiment, everything looked fantastic. I mean, for 25 generations in a row, they were successfully cloning these mice. Not only that, but the success rate actually started to go up. The researchers must have thought they had cracked the code, that maybe, just maybe, this could go on forever. But right around that 25th generation, after hitting that peak, something started to go wrong. The success rate, which had been so promising, started to dip. It was a slow decline at first, but then it got faster and faster, until finally, at the 58th generation, they just hit a wall. The copying process stopped working completely. And we're not talking about a small dip. The failure was absolutely dramatic. At its best, around the 26th generation, the cloning success rate was over 15%, which for this line of work is actually pretty amazing. But by the time they got to the 57th generation, that rate had dropped to less than 1%. The project had finally reached its biological breaking point. So, the big question is, what on earth happened? How could a process that worked so well for so long just completely fall apart? The investigation kicked off, and right away they had 2 main suspects for why the whole process was grinding to a halt. You can think about it like this. Was the problem with the computer's software or its hardware? Suspect number 1 was epigenetic errors. These aren't changes to the DNA code itself. They're more like glitches in how the cells read that code. Faulty software, basically. Suspect number 2 was the hardware itself. Actual genetic mutations, real damage piling up in the DNA over time. Well, it didn't take them long to rule out the first suspect. The scientists had always seen certain quirks in the clones, like abnormally large placentas, but here's the thing. Those problems weren't getting any worse from one generation to the next. The software glitches were there, but they weren't building up, so the real problem had to be something deeper. And that's when they found it, the key piece of evidence. The failure was not in the instructions, it was in the blueprint itself. The DNA was literally, slowly but surely falling apart, a process of genetic decay. You know, there's actually a name for this. It's an evolutionary theory called Muller's ratchet. The basic idea is that any time you have reproduction without any genetic mixing, like cloning, harmful mutations just pile up. Each new generation gets all the mistakes from the last one, plus some of its own. It's like a ratchet that can only click one way, towards more and more errors, until the whole system just breaks down. And the researchers put a number to this decay. It's pretty stunning. With every single new generation of clones, on average, almost 70 new mutations popped up in the DNA. It was like a clock steadily ticking down to failure with each new copy. In those early successful generations, the mutations were pretty minor, but after generation 25, the errors became catastrophic.
We're talking massive structural damage, like losing an entire chromosome or chunks of chromosomes breaking off and sticking to the wrong place. This was the exact breakdown that Muller's ratchet predicted. But just as the experiment showed a hard limit, it also uncovered something amazing, a kind of biological escape hatch, nature's very own reset button. So, in this absolutely brilliant twist, the researchers took some of those female clones from the very late generations, the ones whose DNA was riddled with errors, and they let them mate naturally with healthy males. And the result was just wow! Most of the embryos didn't make it, but a few of them were renormalized, perfectly healthy. The numbers really tell the whole story. The late generation clones could barely have babies, averaging just over 2 pups, but their offspring, the ones born the old-fashioned way, had their fertility totally restored. They were having healthy litters of 7 pups on average. What this all pointed to was a powerful conclusion. Sexual reproduction was acting like a genetic cleaning crew. The process of making eggs and mixing genes with a partner was somehow filtering out and repairing decades of accumulated damage. It's an incredible demonstration of why so many creatures on Earth rely on it. So, after this massive 20-year experiment, what's the big picture? What's the lesson that nature is trying to teach us here? First, this experiment is basically the first real concrete proof in a mammal that Muller's ratchet is not just a theory, it's a hard and fast biological rule. It also sends a clear message that while cloning is a cool tool, it can't save an endangered species all on its own, not forever. You need genetic diversity. And, ultimately, it's just this powerful reminder that sexual reproduction is not just about making babies with new traits. It's a vital maintenance system for keeping life's code from falling apart. Which kind of leaves us with one final big idea to think about. By building this inevitable decay into copying, you have to wonder, is nature sort of protecting itself? Is it preventing a world of perfect, identical copies that could be wiped out by a single threat? It really seems like the errors aren't just a bug, they're a fundamental feature of nature's grand design. And one last thing before we wrap up. While reading this study, I kept thinking about a sci-fi show that some of you hardcore fans might know, Stargate SG-1. I don't want to give away any spoilers, but there's an alien race in that show dealing with this exact
same
cloning problem. And honestly, that's one of the things I love most about great science fiction. It gets the science right before the science does. So if this topic reminded you of any other sci-fi movies or shows, let me know in the comments below. I'd love to hear your thoughts. And what do you think? Could cloning ever overcome these biological limits? Share your thoughts in the comments below. Fresh Science is here to bring you the most exciting breakthroughs from the edge of discovery. If you want to stay ahead of the latest news in science, make sure to hit that subscribe button and join our community. You can find all the scientific sources used for this video in the description below. Keep exploring, keep questioning, and keep learning. Stay curious and stay fresh.